Hello students, welcome back to my channel. I'm uploading this video after a long time and this is a much requested video. So basically today I will discuss about the Icse previous year's questions from the chapter Absorption by Roots. So I hope you will like this video because many students have requested me to make a video on this topic. So without wasting time, let's get started with the video. Now, the first question is, see, a diagram has been given. The question says that the diagram given below, this diagram, I'm talking about this diagram, this represents a plant cell. When now the plant cell is being placed in a strong sugar solution. Okay, so this diagram represents a plant cell after being placed in a strong sugar solution. Okay, now you have to study the diagram and answer the questions. What are the questions? First, look at the picture or the diagram very carefully. First question is, what is the stage of the cell shown in the diagram? So first you have to tell about the state of the cell that is shown in the diagram. So let's see what is the state. The state of the cell is plasmolized. Okay. Now what happens when a plant cell is placed in a strong sugar solution? What happens? Since it's a strong sugar solution, so the concentration of water molecules will be less in that sugar solution. So from the cell, the water molecules will come out. So exosmosis will occur, right? And in this state, what we call it as plasmolized cell. So the state of the cell is plasmolized. Now the next question is, name the structure that acts as a selectively permeable membrane. So in the diagram, which structure acts as a selectively permeable membrane, that is semi-permeable membrane? Of course, it is the cell membrane which acts as a selectively permeable membrane or the semi-permeable membrane. That means it will select which molecules will be able to enter or come outside from the cell, right? Now, what is the third question? Third question is, you have to label the parts which are numbered from 1 to 4. See, in the diagram, numbering is there, right? 1, 2, 3, 4. These 4 labelings are there. So let's see. Now, first one is the cell wall. Second one is, of course, the strong sugar solution. Third one is the cell membrane. And fourth one is the nucleus of the cell. Clear? So you have to just label it. You have to mention the numbers, put a dash or whatever you feel like. Just you have to write down the proper name of that labeled parts. Now come to the fourth question. How can the above cell be brought back to its original condition? And you have to mention the scientific term for the recovery of the cell. Now in the question it has been said that the plant cell is placed in a strong sugar solution. Now the state of the cell, you know, it is plasmolized. So how you can just brought back to the original condition of the cell? And you have to mention the scientific term for the recovery of the cell. So of course you will put the cell in the hypotonic solution. You will put the cell into the hypotonic solution or water. That means hypotonic solution means those solution where the concentration of water molecules or solvent molecules is greater than that of the solute. So you can place that cell, this plant cell, into water or inside any hypotonic solution. Then the plant cell will be reversed back into its original state. And what is the scientific term? It will be deplasmolysis. If the current state is plasmolysed or plasmolysis, if it's that, then of course the recovery of the cell will be deplasmolysis. Clear? Now what is the fifth question? You have to state any two features of the above plant cell which is not present in animal cell. So basically differentiation has been asked to you. Okay, so what will be the answer? It will be cell wall and vacuole. So cell wall is present only in plant cell, it is not present in animal cell, and large vacuoles are present in plant cell. Very small, few very small vacuoles are present, but those vacuoles are so small in animal cells that they are negligible. So you can consider that vacuoles are present in plant cell which are large enough, but in animal cells they are not present. Okay, so this is all about the first question that has come in Icse. I hope this first question is absolutely clear to you all. Now the next question. Here a figure has been given which is basically a diagrammatic representation of the part of a cross section of the root in the root hair zone. You have to study this diagram and you have to answer the following questions. So first you have to study the diagram thoroughly. First is you have to label the parts. You have to label the parts. Those parts that is from 1 to 4. Okay. Now 1 is root hair, 2 is soil water, 3 is xylem vessels, and 4 is cortical cells. Just once again look at the parts. Here is the 1. So it's root hair. Number 2, soil water. Number 3, xylem vessels. Number 4, cortical cells. Clear? Next, which is the process that enables the passage of water from soil into the root hair? So you have to just mention the process which actually help the water to pass into the root hair from the soil, because you know that water is present in the soil. So what is the process which basically enables that water to pass from the soil into the root hair? So of course it is the osmosis. Through cell to cell osmosis this is possible. Next question. You have to name the pressure that is responsible for the movement of water in the direction indicated by arrows, and you have to define it. So see, the water is moving inside the root hair from cell to cell. So what is the pressure? It is osmotic pressure, and you have to define it. Remember that. So the pressure that is exerted by a solution so as to prevent the entry of water molecules through the semi-permeable membrane, that is the osmotic pressure. So it is that pressure which basically is exerted by a solution. Why? So as to prevent the entry of water molecules, obviously through a semi-permeable membrane. What is the fourth question? See, here it has been mentioned, due to an excess of this pressure, that means the osmotic pressure, sometimes what happens, drops of water are found along the leaf margins of some plants. Especially this happens in the early mornings, like we say dewdrops at that time. So what is the phenomenon called? It is very easy. You all know that it is guttation. If you haven't yet watched my previous videos on absorption by roots, just go to my channel and watch those videos first, so you will be able to understand it clearly. And the fifth question is, you have to draw a label diagram of the root hair cell as it would appear if an excess of fertilizer is added to the soil close to it. So you have to draw a root hair cell where in the soil excess of fertilizer has been added. So what will happen to the root hair? What will be the condition? See, there are fertilizers in the soil and this is the root hair, right? What will happen? It will shrink. It will shrink due to plasmolysis. It will shrink. Water molecules will come outside because whenever you are adding excess fertilizers in the soil, it has become hypertonic. So water molecules will come outside of the cell or root hair, whatever. So shrunken cell membrane you will get of the root hair. Clear? I hope the second question is also clear to you all. Now come to the third question. A candidate, in order to study the process of osmosis, has taken 3 potato cubes. Remember, I explained about the potato osmoscope. So, a candidate, so as to study how this osmosis occurs, that candidate has taken 3 potato cubes, 1, 2, and 3. And that candidate has put them in 3 different beakers that also contain 3 different solutions. Now, after 24 hours, in the first beaker, if I consider this as the first beaker, the potato cube increased in size. Okay. In the second beaker, what happens? The potato cube decreased in size, just the opposite. And in the third beaker, there was no change in the size of the potato cube. Just same as it is, remains same. So the following diagram has been represented so as to show you the result of the same experiment. Okay. Then what is the question? Give the technical terms of the solutions used in beakers 1, 2, and 3. Now, of course, in beaker 1, the solution is hypotonic in nature. That is why the size of the potato cube has increased, because water from the beaker, water molecules from the beaker is going towards the potato cube, inside the potato cube. So endosmosis is occurring. That is why the size of the potato cube is increased. So it is obviously hypotonic solution. Second one, potato cube size has been decreased. So it is hypertonic solution. So water molecules has come out from the potato cube into the beaker. So exosmosis has occurred. And in the third one, there is no change in the size of the potato cube. So of course, it's isotonic solution. Concentration of solute and concentration of solvent is same in this type of solution. So there is no change in the size of the potato cube, right? Now, the second question. In beaker 3, the size of the potato cube remains the same. You have to explain the reason in brief. I already explained this. In beaker 3, what happens? The concentration of the potato cube and the medium is same. So there is no osmosis taking place. So as a result, what is happening? The size of the potato cube remains the same because it is isotonic solution. Third question is, you have to write the specific feature of the cell sap of the root hairs, which helps in absorption of water. Now, the concentration of the cell sap is higher in the root hair, right? Inside the root hair, the cell sap that is present, its concentration is higher as compared to the soil water. So as a result, what happens? Water from the soil enters into the root hair due to endosmosis. This basically helps in the absorption. This facilitates the absorption of water, right? Since the cell sap that is present inside the root hair, it is higher in concentration. So what will happen? There is less of water molecules. So water molecules that are present in soil, they will enter inside the root hair, right? So basically, this scientific feature of the cell sap of the root hair is responsible for facilitating the absorption of the water, right? Next question is, what is osmosis? We have already discussed. Osmosis is the process of flow of solvent molecules from its region of higher concentration to its region of lower concentration. That means, and of course, through a semi-permeable membrane. That means osmosis is that process where water molecules will move. Okay, we can explain this process from the point of view of the solvent or water molecules. So these solvent molecules or water molecules will move from their region of higher concentration to their region of lower concentration. That means they will move from that region where their concentration is higher than to that region where their concentration is lower. Clear? And fifth question is, how does a cell wall and a cell membrane differ in their permeability? Right? So basically, it's a difference. Cell wall is freely permeable. It will allow every molecules to go inside or come outside of the cell. But cell membrane is semi-permeable. It will selectively permit. Clear? I hope this question is also clear to you all. Now the next question. A figure has been given which shows the epidermal cells of an onion bulb. This cell was then transferred to a drop of sugar solution. Okay. This is basically an epidermal cell of an onion bulb, and then this particular cell has been transferred to a drop of sugar solution. First, you have to draw a well-leveled diagram of this epidermal cell when, as it would appear after immersion in a strong sugar solution. See what will happen. This is the diagram. If that cell is dipped into the strong sugar solution, then there will be shrinkage of cytoplasm. Exosmosis will occur. Water molecules will come outside of the cell into the sugar solution. Exosmosis will occur, so protoplasm or cytoplasm, they will shrink. Plasmolysis will occur. Okay. Second question, what scientific term is used for the changes that is shown in the question number 1 above? Of course, plasmolysis, I have told you. The technical term is plasmolysis. Question number 3. What should be done to restore the cell back to its original condition? Same question has been asked repeatedly. All these are previous year's questions. So now you can understand that same type of questions are coming, just with different examples. So what will you do so as to restore back the original condition? Of course, you will place it immediately into the hypotonic solution or water. Right? I've already discussed that. And what is the scientific term for the recovery of the cell? It is deplasmolysis. Right? Again, the same question. Define the term osmosis. You have to define the term osmosis. So, I have already discussed osmosis is the process where there is a movement of solvent molecules or water molecules from their region of higher concentration to their region of lower concentration, of course through semi-permeable membrane. Clear? So, all these questions are repeating. They are repetitive in nature. Same type of question will come year after year. You have to just practice this type of questions. I hope all these type of questions that I have discussed today in this video is absolutely clear to you all. Even if you have any doubts after that, then do comment. I will try to clear your doubts. So this is all for today. I think you have understood all the topics that I have covered today. If you think that my videos are informative, then please like my videos. You can comment and share my videos with your friends. And of course, don't forget to subscribe to my channel. I'll be back with a new video very soon. Till then, stay tuned to my channel. Thank you.